So the study we presented here was AMG 330, a CD33 T-cell recruiting antibody construct in relapsed refractory AML. So this construct is sort of the sister molecule of blinatumomab, targeting CD33, which is ubiquitously expressed in AML in this early phase 1 trial. The expected problem with T-cell recruiting antibody construct is cytokine release. So we started with a very low continuous infusion, as these BiTE molecules are very small and have a restricted half-life of less than 2 hours. So we eventually encountered cytokine release syndrome, and mitigating this, we implemented dose steps to increase. So currently we have 2 dose steps and extending the time of infusion of the AMG 330 to now 28 days. So as I said, side effects have been manageable and were expected, and mainly are cytokine release. Contrast to the T-cell recruiting antibody constructs in the ALL setting, we did not encounter a significant number of neurotoxicity. And for efficacy, clearly this is hard to say in a phase 1 trial, but now in the higher doses, we have observed several complete remissions or incomplete remissions. So we are optimistic that we eventually now achieve the concentration, and we are still moving up where we'll see efficacy. But clearly a lot of patients have been treated where we did not observe a clinical benefit. So next is we're gonna escalate until we have achieved our final dose, and then there's gonna be an extension phase to see for efficacy. But I think very similar to blinatumomab, we have to change the clinical setting. So I think T-cell recruiting antibodies are most beneficial in the MRD setting. So we have been discussing to integrate also patients who are MRD positive, and this could serve as a bridge to transplant, or we can see if MRD conversion is taking place in a setting where the T-cell compartment is more fit, and probably the antibody is going to work much better than in the very advanced setting. So these patients had 4 or more treatment lines prior to the integration to the trial. So clearly very heavily pretreated patients where T-cell exhaustion, T-cell dysfunction might be a problem. So I think these are exciting times, and we'll see how the success stories of BiTEs and CAR T-cells in the lymphoid settings are going to translate into the myeloid setting. There's several trials that are reporting with other target antigens and a few trials on CAR T-cells, but clearly it's early days, and identifying a suitable target antigen in AML is much more difficult than in the lymphoid setting.